Now we're finally getting to cell biology. I'm so happy we're finally here. Okay, so to save us on time, and again, I did warn you about this. This is a prototypical human cell. What does it mean by that? This disclaimer in the book is very important. So this isn't, if you look at any given cell in the human body, it probably won't look like this picture. But what is it showing you? It's just showing you all the possible types of organelles and substructures you can find within a cell. So again, a cell won't typically look like this in your body, but it's just showing you all the basic organs of the cells, aka the organelles. And this is the Martini version, and it has a little more information. I think the OpenStax version is a little more clean. So again, if you want a clear picture of what the different types are, OpenStax, and then Martini has more details and a better, also has little summaries of their general functions. So this is why I want everyone to do at home. Rather than me pointing out and saying, this is a mitochondria, this is a peroxisome, this is the Golgi apparatus. I want you to study these on your own time. The future homework will have these as well. But you should be able to identify the organelles based on these pictures. Everyone has the slides. And know general functions based on the book. Again, don't go too deep in depth for any of these organelles. Like if you're talking about calcium flux or ryanodine receptors or something like that, you're studying way beyond the scope of this class. Just go with what's in our book. Again, we're just starting out very general. Okay, so things that cells. Why do we talk about cells? Well, cells, and spoiler for the homework, is that cells are living. So cells can need nutrients. They need to maintain themselves. If they don't get nutrients, cells can die. Chemicals can't technically die, right? Because again, they can maintain their structures, and you can have a vial of a chemical, store it indefinitely. And atoms don't die either. Well, unless you're talking about radioactive decay, but that's not technically dying. So cells are the smallest living unit in your body. So these are building blocks. Your body is made out of cells and extracellular matrix, which we'll get into chapter 4. But cells are the basic living units of your body. So they are building blocks, and it's kind of like this. I like to teach my little nieces and nephews were still in elementary school. I like to use this. I told them like cells are like this in Minecraft, right? So again, it's like and they try to get me into it, and I was like, okay, this is a little too addicting. I'm wasting too much time. But the thing is that it's kind of amazing. So you have building blocks, and this elaborate structure right here, all made from building blocks, right? So just like your body is made out of cells, same with all these structures in Minecraft. You can build these amazing structures. And the thing is that, why do I use this kind of analogy? The thing is that not all the building blocks are the same. So not all the cells in your body are one and the same. You're not just made out of one type of cell. You have many different cells with many different properties. So then you also have logic gates. I know it's pretty amazing. So the thing is that sometimes having different combination of those blocks can cause different activities or different functions as well. Same with your body. So your organs and tissues, they're not just made out of just one type of cell. Sometimes you have multiple types of cells that work together to carry out the overall function of that tissue or organ. So the thing is, okay, so let's talk about two broad categories of cells. So they're either a sex cell or a somatic cell. So sex cells, what we have here, these are germ cells or gametes. So these are reproductive cells. In female, that's the egg or the ovum. In male, that's the sperm or spermatozoon. And then somatic cells, pretty much everything else that's not a sex cell, that's not a reproductive cell. So soma is a term meaning the body. So when you hear that term soma, that refers to something involving the body. Of course, that's pretty much everything we're talking about, right? So sex and somatic cells, those are two jobs. Again, this is like pretty much everything but sex cells. Yeah, there's no overlap. They're mutually exclusive. All right, so basically, remember, your cells are living. Therefore, they need things like nutrients, water, electrolytes. And the thing is that just like how you're supposed to drink in things, you need energy, you need nutrients, is that all your body needs to do? No, again, it's like what happens to your food eventually? You kind of need to get rid of all these wastes and all these byproducts like carbon dioxide. So this is why we produce urine and feces. You have to get rid of excess waste and metabolites. So how does this happen? So the thing is that the plasma membrane. So the plasma membrane, this is a sheet that envelops every single cell, but it's more than just a simple sheet. It's actually a very important barrier in determining what gets into your cells, what gets out of the cells, how much gets into your cells. So it's actually a very important first barrier in protecting your cells, but also allowing things that it needs, like nutrients and water in, but also important into preventing things like toxins and environmental hazards from getting inside of your cells. So it's actually a very important barrier. So you have things called phospholipids. We did cover a bit of this in our last lecture. So again, phosphate group right here. So what we have is a phosphate group that's linked to two lipids. So this is why you call it phospholipid. And remember that phospholipids are that A word we talk about. So it has a hydrophobic side, that's the fatty acid side. Remember, fatty acids don't dissolve well in water, whereas the head right here with the phospholipid or the phosphate group attached to the lipids, this is the hydrophilic side. So this is the example from your open stats book. So you can see the phosphate and the glycerol right here. So they're amphipathic in that they can both have a side that's they have a side that's hydrophilic, and they have a side that's hydrophobic. So they have properties of both hydrophobic and hydrophilic properties. So what happens if you throw a bunch of phospholipids into a solution? Well, the hydrophilic head will associate with the water-based solution. So that's what we have here. But what do you notice about the hydrophobic tails? Notice that they're all pointing the same direction away from that aqueous solution. And again, that aqueous solution, the water has a hard time grabbing onto these tails because they don't really have a strong electrostatic charge. The thing about your cells in the plasma membrane is that not only is there one side, the outside of your cells is mostly aqueous, but the inside of your cells is also aqueous, in other words, water-based. So you have both the hydrophobic side on the top facing the outside, and you also have an aqueous solution within your cells. So what you have eventually is that, but this is why we call it a phospholipid bilayer. It's not just one layer of phospholipids, you actually have two layers. So right here is kind of like the border between the inner and the outer layer. So again, why the hydrophilic heads like to go toward the areas that have water. So water likes any sort of positive or negative attraction. So thanks to these phosphate groups, these slightly negatively charged phosphate groups, this is what allows water to grab onto that. So again, the outside of your cells is water-based, and the inside of your cells is also water-based. So again, this is why you actually have these phospholipid heads pointing toward the outside and inside of your cells. But so this part of the membrane is hydrophilic, the outer and inner parts. But this part right here, this is what is sometimes referred to as the hydrophobic core. So because of all those fatty acid tails being poking toward the inside, and actually in this image, you can see small cholesterol molecules as well. So the inside of a plasma membrane actually is a hydrophobic layer. So this is what we have, kind of like a hydrophobic sandwich. Hydrophobic on the inside, hydrophilic on the outer parts of that plasma membrane. So what is a membrane? What is this membrane word I'm talking about? So a membrane in general is a flat, broad structure. So it's just like a bed sheet or something. So it's something that's flat, very broad, very wide. Now, the thing is that this is also where you have to be kind of careful, and I'll try to be as precise as possible. So today I'm talking about the cell membrane. So this is the membrane that surrounds all your cells, also known as your plasma membrane. When we get to tissues, there is another type of membrane that is referring like the synovial membrane or the cutaneous membrane. That's not referring to the cell membrane. So again, when I'm talking about this lecture, I'm talking about the cell membrane. So that's a tough thing about A&P. Sometimes you see the same word, sometimes it will just say membrane, but just by context, you kind of have to know if it's a cell membrane or a tissue membrane or some other membrane. So then we have to talk about permeability. So this is a big SAT word. So what does permeable mean? So basically, it's the ease at which something can pass through something. So basically how porous it is, how easily something can cross from one side of a membrane to the other side of the membrane. This is permeability. So, or things that if you have something that's impermeable, that means nothing can really cross it. So if it's an impermeable membrane, nothing can get through. Everything stops, no matter what. Then you have freely permeable. That is like having something, it's pretty much like a membrane, but it has huge holes in it that anything can pass through. So anything can cross. So everything has to, anything and everything has to cross for it to be freely permeable. Whereas this middle ground, selectively permeable. So that means it selects what can cross. So some things can cross through, but not everything can cross through. So that's like selectively permeable membrane. So impermeable and freely permeable, these are extremes, and anything in between, that's selectively permeable. As long as something can get across, but not all things can get across, it is selectively permeable. But what about the plasma membrane? So is it impermeable, freely permeable, or selectively permeable? Well, let's investigate. So here we have a bunch of water molecules again. The inside of your cells and outside of your cells is also water-based. So we have all these water molecules right here. But things that water molecules, technically, they have a hard time getting through this hydrophobic core right here. So, but things that they're so small enough that they can actually squeeze between the phospholipids and end up on the other side. And this kind of happens both ways. So the water molecules, they do have a tough time passing through the hydrophobic core, but they are small enough that they can squeeze through. So small molecules and ions, or actually not ions, small molecules can pass through the plasma membrane. But there's also special proteins called aquaporins, and I think this is what it's showing in this picture right here. So aquaporins, aqua referring to water, porins sounding like pore, right? So a pore is basically some sort of hole in the structure. So here's a hole that allows water to go through. So the funny thing is, especially when you get to the kidney, the aquaporins are found all over in the place in the kidney. So actually, even though water has a hard time getting through the hydrophobic core of the plasma membrane itself, actually when you open all of these little holes, all these little channels that allow water through, water can also flow through this way as well. So water and plasma membrane can cross this way. Now, this funny thing about this, here we have a cartoon of sugar molecules, or glucose molecules in this case. So think about glucose, it has a hard time passing through the heart. It can't, well, think about with aquaporins, it only allows water through, so it can't use that. But think about glucose, is compared to a water molecule, it's relatively huge, and also has a hard time squeezing through these phospholipids. Why? Because glucose has partial positive and negative charge. Even though the overall molecule does not have an overall charge, there are pockets of it that have positive and negative charges. So things that makes it harder for it to pass through this kind of like a gauntlet of phospholipids and these hydrophobic fatty acid tails in these phospholipids. So glucose and large molecules have a hard time passing through the hydrophobic core of the plasma membrane. So without additional help, glucose cannot pass through the plasma membrane. Now let's talk about ion channels. So the thing is that sodium by itself cannot go through aquaporins, and also due to its high charge, again, remember water and things like ions, they dissolve easily in water because they have a charge. But compared to something like a hydrocarbon chain on a fatty acid, they have a hard time passing through that because there's no real attraction between a charged ion and the hydrophobic tails of a phospholipid. So this is why ions are typically kept out and kept from crossing through a hydrophobic core of a plasma membrane. Now when you have an ion channel, what does that do? So here we have our ions back for more. But when you have an ion channel, it's a special type of protein that allows the passageway for ions to go from one side of a membrane to another. But again, by themselves, without the ion channels, these ions cannot cross from one side to the other, so they need a little help. So in summary, what can pass through the lipid bilayer of the plasma membrane? Well, again, what can freely pass? So things that are very small. So things like oxygen, carbon dioxide, which are technically nonpolar and technically, if you had to classify them as hydrophobic or hydrophilic, they're technically hydrophobic, but they're so small they can easily pass through. So things like water, they're technically hydrophilic. So then, but things that, why isn't it kept out by the, well, it is kept out to an extent by the lipid-based inner core of a plasma membrane, but so small it kind of has a free pass. And same with ethanol, which is also known as drinking alcohol. And then you have nonpolar molecules. So things like, actually oxygen and carbon dioxide are nonpolar. They can pass, not due to being small and being able to freely diffuse throughout the lipid bilayer. So most sterols and steroids can pass through the lipid bilayer, so they kind of like have a carte blanche to just go anywhere that has a hydrophobic core. And other hydrophobic molecules and drugs. So if you're planning on going to pharmacy, nursing, or medicine, so this is also important to know because if you have a drug that's hydrophilic versus one that's hydrophobic, something that's hydrophobic, it has a hard time dissolving in water, but once you get into the system, has a relatively easy time passing through the plasma membrane versus something that's very charged, very big, and more hydrophilic. Now, large molecules. So again, if something's way bigger than a phospholipid, it's going to have a hard time squeezing between these phospholipids. And highly polar compounds, again, this is why it's important to know that the inner hydrophobic core of the lipid bilayer exists, because these highly polar compounds have a hard time passing through hydrophobic regions. So things like ions, they also have strong electrostatic charge, positive or negative. And okay, please work this time, top hat. So based on everything we just covered, what in your best estimation, so what would you classify the plasma membrane as? All right, let's see how everyone did. So yes, most of you are correct that it is semi-permeable. Why? Because remember, without the addition of ion channels or without the addition of things like glucose transporters, things like glucose, things like ions, just with the plain plasma membrane and no other proteins, nothing else helping it get across, it has a hard time getting it can't really pass through. But things like water, it can freely pass across the plasma membrane, along with things like oxygen, carbon dioxide, and ethanol. All right, so back to our lecture. So again, I summarized all of these. So the thing is that there are the membrane transport basically refers to how do things get from one side of a plasma membrane to the other. So there are two broad categories of membrane transport. It's either passive transport. In other words, this doesn't require any additional energy. So if you just have a membrane, you just have whatever is being transported. This transport happens by itself. No energy, no ATP, no additional nutrients needs to be added. Whereas active transport, it requires additional energy that's stored up in some form. Sometimes it's ATP, sometimes it's having more ions on one side versus the other. So membrane transport falls into either of two broad categories. So these are the types, and we'll try to cover the passive transport this lecture. All right, so this is my unofficial rule of thumb for physiology. So in terms of passive transport, thing is your body loves to maintain homeostasis, right? So thing is that your body, this is what I call the high to low rule. In physiology, things tend to move from high to low. So high to low concentrations, things if you just have a high concentration on one side, low concentration on the other side, things tend to flow to the low concentration. If you have high pressure on one side, low pressure on the other side, things tend to flow to the low pressure. So this is actually carries you a lot through a lot of these physiology concepts, especially when you get to PHYL 142. This concept actually applies to many things. So the thing is that osmosis is a little special. So people tend to get confused on that. But let's first talk about something that's more basic. So diffusion. So what happens is like diffusion, say you have all these molecules and this is an enclosed space, and then you just release all of these molecules in one corner of the space. So I remember like when I was in orchestra during in high school, then some kid, they pranked and they took a stink bomb and they cracked it on one side of the room. So what was funny is that stink sulfur odor, that rotten egg odor, started to travel from one side of the room to the other. So it was really funny because you could tell like the violins, they started giggling and some people were gagging, then the violas, then the cellos, and finally it reached the double basses who finally smelled it. So the thing is that diffusion, what happens? Things tend to move from high to low concentrations. Same with gas particles, same with anything that can freely move across a space. So here we have a high concentration of these gas molecules right here. But when you let them loose, eventually they'll try to spread out. They'll try to do the social distancing thing, right? So diffusing, if you have an unlimited space, things that molecules like to have their own space as well. So if you put them all into one corner, eventually over time, without any additional barriers, they'll spread out till they're roughly equidistant from each other. They're still moving, but they will try to spread out to a more diffuse area. Now it's kind of like people. Like say you're in two areas. Here we have an equal amount of people. But say this part gets really crowded, and actually this is what would happen in real life now. Where would you rather be, in this part right here or this part right here? Well, people, if they have two spaces and they're really concentrated in one area versus this area with fewer people, they're probably going to spread out as well. So molecules don't like to be crowded either. Neither do people, unless you really want to catch some stuff. Then people tend to spread out over an area if they're really bunched into one area, right? So molecules are kind of like you. They like their personal space. They like their boundaries. So diffusion moves things from high to low. And you'll see me using this notation a lot for shorthand. So whenever I put something within a single bracket like this refers to concentration. So I'm saying it moves from a high concentration to a low concentration. I'm using it a little, like, not unconventionally this way, but I'm just, FYI, this is my shorthand. Okay, so we talked about diffusion. So basically from a high concentration of particles to a low concentration of particles. Now then, what's facilitated diffusion? So it is a subtype of diffusion. Just like, again, it's not a mutually exclusive thing, but it has a certain twist. So facilitated diffusion, here we have glucose. And remember I said, like, by itself, without any additional proteins, glucose cannot really pass through the plasma membrane. But there's something called facilitated diffusion. So here we have glucose again. Here we have the plasma membrane, but now we have a special protein, a carrier protein. So what happens is that glucose is able to fit into the little pocket of this carrier protein. And this carrier protein allows glucose to go from one side to the other side. So it kind of shuttles it around. So the thing is that without it, glucose can't pass through. But thanks to this carrier protein that helps to ferry across, glucose can pass through the other side. So it's kind of like a turnstile. So why do I use that term? Well, it doesn't really require thing with the turnstile. Is it powered by electricity? No, people just power it itself, right? So things that facilitate diffusion. Say you have this space, but it has a big impermeable wall between it. So can these people get from one side to the other, even though they're all crowded in this space? No, because again, there's no door. But say you have a turnstile, what can these people do? Now they can move from one side to the other and spread out. This is kind of like how the facilitated diffusion works. So it still involves high to low, but just due to this difference, and this is what we call a concentration gradient. So gradient referring to a more technical term, because the thing is that it's not always complete, like, okay, night and day difference between two different solutions. There's usually some sort of like a kind of continuum. But don't worry, when you hear that term concentration gradient, when you're starting out, it's easier to think of it as concentration difference. Again, it's not quite exact, but if you're not familiar with gradients yet, and if you're taking Calculus 3, you'll be very familiar with gradients. But what I'm getting at is that there's a concentration difference. But due to that concentration difference, these transporters, they don't require energy. It's just due to this difference that drives the movement of particles from one side to the other. So it still moves from high to low concentration. It still is diffusion. It still moves something from high to low concentration. But instead of having just a pure plasma membrane and a membrane barrier, then it actually has something helping out, typically a protein. So this is involves particles that cannot pass through by itself. Still allows, and things that these transport proteins like that glucose carrier molecule, this allows the movement. But by itself, if a particle or some sort of solute that undergoes facilitated diffusion, it cannot move by itself. That's the rub for there. Okay, so let's get to osmosis. Very, very important concept. And this is one of the big hurdles, not only in general biology, but it's just it's one of the things I noticed that if you kind of it's hard to get at first, but once you get over this hurdle, it kind of just really makes sense. So solutes and solvents. So remember, we did cover this in previous lectures. So solvent is a substance used to dissolve other chemicals. Solutes are what being dissolved in the solvent. So solution, this is a mixture of a solute dissolved in the solvent. Okay, so let's hope top hat works. Next question. Okay, let's see how people did. So let's see. So in this solution, just talking about plain salt water, most of you said NaCl is the solute, water is the solvent. And the majority of you are correct. So again, solute is the thing being dissolved, and solvent, that's the thing doing the dissolving. So what you have with salt water, you have salt being dissolved in water. So again, water is the main solvent, solvent in the human body. All right, in case you missed that, mulligan. So we're talking about basic simple syrup, in other words, sugar water. Which is the solvent, which is the solution? Okay, let's see what people said. So the majority of you said that water is the solvent, simple syrup is the solution, and then second said that Water is a solvent, sucrose is the solution. And which is correct? The first choice is correct. Again, solution refers to the overall mixture. So the people who selected D, sucrose is not the solution, it's the solute. So again, this is where it gets very tricky. Again, they all sound very similar, but they refer to different parts of a solution. Again, the solute, ending with the E, that is the substance being dissolved. That's solid being dissolved in some sort of liquid. Water is the solvent. Again, the solvent is the liquid doing the dissolving. And the overall solution, that's the mixture of the solute plus solvent. Okay. All right, so back to our lecture. Okay, so we talked about membrane permeability. So impermeable, freely permeable, and selectively permeable, which is our plasma membrane. Some, but not all molecules can cross. So osmosis refers to the diffusion of water, and this is where the high to low rule still applies. So often people start to confuse it, okay, is it from actually a high concentration to low concentration? Or it's like, oh, but what about water? So doesn't that technically go from high concentration to low concentration? And you're correct. The thing is that water molecules move from a high water concentration to a low water concentration. Or some people like to think of it as that the high concentration of solutes likes to draw in water. Whichever works in your mind, it kind of works. But it's one of those things that is kind of a mental hurdle. So things that the selectively permeable, this is why you have to understand this. If you don't understand selectively permeable, you have a hard time understanding osmosis. So things that this is a plasma membrane. Again, without the addition of additional ion channels, things that sodium has a hard time passing through the plasma membrane due to its charge. But water molecules, due to their small size, they can freely pass through the plasma membrane to an extent. But again, because they can squeeze through the phospholipids, they can go from one side to the other. So what we have here is that it's selectively permeable because, as we saw, sodium has a hard time passing through, but water has an easy time passing through compared to sodium. So again, sodium cannot pass through. So with osmosis, how does that relate to osmosis? So things that solute concentration drives osmosis, but there's a twist. So things that nature likes equilibrium, and it's not just about homeostasis. Nature doesn't like it when you have you have more things piled up on one side and less on the other. And if there's any ability for it to pile from the high concentration to the low concentration, nature is going to push it that way. So things that osmosis tries to balance out the concentration. If there is a concentration difference between two solutions and there's a membrane between them, then nature tries to make the concentrations equal, tries its best. So things that solutes and water attract each other. And why is that? So things that, remember, water, due to its partial positive and negative charge, any sort of charged solute is going to attract each other. So it's not just one way. They do attract each other. And this is what we have here this is what we covered last lecture. So again, remember, those charges attract, so negative attracts positive and positive attracts negative and vice versa. So this is how these water molecules are able to dissolve these ionic compounds, or any compound with the charge or enough charge. So here's our semi-permeable membrane, and these charged molecules have a hard time getting through that hydrophobic core. So the thing is that in this case, we're just showing sodium ions, but these sodium ions, they're kind of trapped. They can't really move across this semi-permeable membrane. But what can pass through a plasma membrane? So the thing is that if solutes can't move, then water moves in terms of the plasma membrane. So remember, our solutes are kind of trapped. The sodium is trapped, but water can move from one side to the other. And why is it going to move this way? Well, notice that in this figure, in this cartoon, I'm only showing 6 water molecules on both sides. But notice that you have the water to sodium ratio. It's like twice the ratio of sodium. But over here, you have fewer water molecules compared to a large concentration of sodium here. So this is very dilute, and this is very concentrated. So what's going to happen is that the water is going to try to balance it out. So now we have an equal amount of water and sodium molecules in this example here. Again, you're not going to find this in your cells. But now we have it balanced out so that there's a more equal amount of water to solute ratio. So the thing is that with osmosis, the overall goal of osmosis is to balance out the concentrations on both sides of a membrane. So it's kind of like people. So again, here is an example. So pretend, like, again, this is pre-social distancing and lockdown days. But here we have an equal amount of people, and I'm using them as analogies to water molecules. So say you have two rooms, and then there's party in one room, again, pre-COVID. So it's low solute concentrations, things like ion salts and like sodium chloride, like glucose, they all contribute to osmosis. They can all dissolve in water. So you have all these yummy, salty, and sugary snacks on one side, but this side has more. So you have an equal amount of people between these two sides, and you have more snacks on this side, more salt, more sugar on this side. So right now, the membrane is impermeable, but what if you open these little doors that only allow the water molecule people through? Which way are they most likely to go? To the part that has fewer snacks that are attractive, or the part that has more snacks? They're probably going to go to the side that has more snacks, right? So this is how water molecules, they find things like glucose, they find things like sodium chloride very attractive. So if there's more on one side of a membrane, that's going to draw more water to that side that has a high solute concentration. All right, so things that in osmosis, this is one of the big tricks. Water can move, for at least osmosis in the human body, water can move freely, but solutes can't. So semi-permeability, this is why you have to understand the difference between impermeable, freely permeable, and semi-permeable. So things that the overall goal of osmosis is to balance out solute concentrations on both sides. So as you saw in that previous party, now people had a kind of equal amount of munchies to people. And then things that water also moves to the side with a higher solute concentration. So again, they didn't like to be in that part with they had some attraction to the relatively fewer goodies on one side, but they moved to the side where that had more attraction, more goodies for them to be attracted to. So osmosis is a high to low special case. So again, we're not talking about concentrations like about the solutes. We're talking about balancing out the concentrations and moving from where there's too much water to where there's relatively less water. So it's still high to low, but with a special twist to it. So this is another way to think of it. Some people get it, and so I think there's two ways to they both refer to the same thing, but it seems that people either understand it one way. It's either that high solute concentrations draw in water, or water moves from in osmosis, water moves from a high water concentration to a low water concentration. But again, do not confuse water concentration with solute concentration. You I really emphasize solutes, solvents, and solutions. All right, so let's end with a few top hat questions. Do you understand osmosis? All right, let's see what people said. So majority of you said that solution B will gain water. And let's see. And then it's spread throughout the other side. So which is correct? Well, solution B will gain water. And why is that? Remember, which has a higher concentration? Again, so the higher concentration, there's more solutes, there's more glucose. You don't have to even worry about the units in this case. It's referring to the same units. There's no math involved. Which is bigger, 200 or 100? So by having twice the amount of solutes, thing is that if water is only allowed through, all those glucose molecules are going to draw in. It's kind of like a tug of war. Who's going to win out? A tug of war with 200 people versus a tug of war with one side having only 100 people. So the 200 is going to have more force and attraction to water. So eventually it'll end up so that you have an equal amount or equal concentration of water or equal concentration of solute and water on both sides. So again, remember, this is one of the overall goals of osmosis. All right, next question. Okay, let's see what people said. So majority of you said that solution B will gain water. And the majority of you are correct. It is the same thing. Which side had more, solution A or solution B? So what we saw is that solution B had a higher concentration of sodium ions. So it's going to go from this side to this side. So again, all those sodium ions attracting water, so it's going to pull it out. Which side has a higher concentration? That's the side that draws in more water. Okay, so last example. So given this solution, what will happen? Okay, let's see what people said. So seems like the majority of you said solution B will gain water again, and the majority of you are correct again. And why is that? So again, even though I said it's pure water, when it's pure water, there's no solutes dissolved in it. So technically that's 0 millimolar in sodium chloride. So which side has the more solutes? Which one is going to draw in more water? Well, without any solutes to really keep it here, water will be attracted from this side with the lower concentration of solute to the higher concentration. So this pure water is going to keep on going down, 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 and it won't quite it will try to dilute the solution B as much as possible, but due to things like air pressure and gravity, yeah, maybe it won't completely drain. But the thing is that it's going to try just through osmosis itself, as much water as possible is trying to cross this side to dilute that really salty or relatively salty concentration. Okay, so again, don't worry if you got it wrong. And again, if you study on osmosis or show up to office hours if you still don't understand. But osmosis, very important. And when we get to next lecture, I'm going to talk about why it's important. But again, enjoy your, now we have our 3-day weekend, our real 3-day weekend. So you have 4 days to kind of let osmosis sink in your brain. So see you back on Wednesday. Don't show up and expect a live telecast on Monday. Please stay safe and enjoy your Labor Day weekend, all right? Okay, take care, everyone. Thank you for all showing up.